rabies. Chances are you've heard of it. But what exactly is rabies? Well, rabies is a disease of the central nervous system caused by a virus in the Rhabdoviridae family named rabies lyssavirus. While it is an entirely preventable disease, rabies is fatal in 99% of cases, and it is responsible for 59,000 human deaths worldwide per year, most of which occur in low-income countries. To understand more about why these deaths occur, let's take a look at the virus itself. This is a rabies virion, a single unit of the virus capable of reproducing in a host cell. It is incredibly small, 75 nanometers wide and 180 nanometers long. To put that into context, a human hair is 75,000 nanometers in diameter, and a human red blood cell is 6,000 nanometers wide. It is composed of RNA that makes up the viral genome and 5 types of proteins, N, M, P, L, and G, that together make up a structure called the nucleocapsid. This is all encased in an envelope taken from a host cell's membrane, with one type of protein, the G protein, extending through the envelope. So, this rabies virion exists, somewhere, floating around. Viruses have no way to move on their own, so it has to be transmitted through direct contact with its new host. In the case of rabies, this transmission happens zoonotically, which is just a fancy way of saying from animals to humans. 90% of human cases worldwide are transmitted from dogs, and rabies is endemic in dog populations in low-income countries, particularly in Africa and Asia. In the United States, bats are the main cause of transmission to humans. In the late stages of a rabies infection, the virus is present in the saliva, so it is transmitted to new hosts through bites or even aerosols that can be caused when a bat flies a little too close to catch a bug. So, you have rabies virus in saliva on a human cell. Before the virus can start causing disease, it has to infect that host cell. This happens when a viral glycoprotein, or G protein, binds to a receptor on the cell membrane. Your cells are covered in receptors that are involved in all sorts of cell-related things, like cell-to-cell communication. Different receptors attach to different molecules, and viruses hijack these receptors in order to get inside the cell. Rabies virus can bind to a number of different receptors. These are just a couple that rabies has been shown to bind to. Once the virus binds to the receptor, the cell mistakenly recognizes it as something that it wants to take up, and envelops the virus through a process called endocytosis. Part of the cellular membrane around the virus pinches off and becomes a vesicle called an endosome, a small compartment within the cell derived from the cell membrane and used for transport of extracellular material. At this point, the endosome is acidified, meaning that hydrogen ions are pumped into the endosome from the cytoplasm, and a change in structure of the G protein causes the viral envelope to fuse with the endosomal membrane. This expels the viral nucleocapsid out of the endosome and into the cytoplasm of the cell, where it can begin replication. The first step in viral replication is the conversion of the viral genome into mRNA, which can be read by ribosomes in the cell and translated into proteins. This process is carried out by the L protein, which is an RNA-dependent RNA polymerase, meaning that it translates RNA into a complementary strand of RNA. This is necessary because the rabies virus genome is made up of negative sense RNA, which cannot code for proteins before being transcribed into its complementary positive sense form. For example, a section of the negative sense RNA may read UGA, GUC, and not lead to any protein synthesis, whereas that corresponding section of the positive sense RNA would read ACU, CAG, and continue synthesis of a protein. The viral genome codes for separate mRNA segments in the order of N, P, M, G, and L. Due to reduced binding of polymerase to genes present farther down the RNA, this order leads to a transcription gradient, where N protein mRNA is transcribed in the greatest amount, and therefore leads to greater expression of N protein, and L protein mRNA is transcribed in the lowest amount, and leads to lowest expression of L protein. Viruses hijack cellular ribosomes to translate viral mRNA into proteins, which happens in the cytoplasm in the case of rabies. Once enough viral protein has been synthesized, the N, P, and L proteins come together in the cytoplasm, along with newly replicated viral genome, forming a ribonucleoprotein complex, or RNP. G protein is transported along membranes of the endoplasmic reticulum to the Golgi body and then to the cell membrane, where it is localized in small clusters. M protein associates with the RMP and G protein embedded in the cell membrane, bringing them together and inducing the cell to undergo exocytosis. Through this process, the cell membrane expels the virus along with the section of the membrane coated in G protein that then becomes the viral envelope. At this point, it is a mature virion, and the virus has completed its life cycle and is ready to infect another cell. Viral reproduction within a host cell generally creates thousands of virions per cell, and the exact number is dependent on a number of factors such as host cell metabolism and host cell type. Rabies viral infection generally begins in muscle tissue, at the site of the bite or scratch. The virus undergoes low levels of replication there until it reaches the peripheral nervous system, where it jumps into peripheral neurons through neuromuscular junctions. Once the virus has reached the nervous system, it travels by retrograde transport through and between neurons until it reaches the brain. Replication and transport in the peripheral nervous system causes no damage to the infected neurons and avoids activation of the immune system by directly inhibiting gene expression and modifying signaling pathways. The time it takes for the virus to reach the central nervous system and start causing symptoms after initial infection is called the incubation period. This typically lasts between 2 to 3 months, but varies by exposure location and viral load and can be anywhere between 1 week and 1 year. Once rabies virus reaches the brain, it undergoes rapid replication and can later spread to the autonomic nervous system and peripheral salivary, adrenal, and lacrimal glands, where it can then be passed on to another host. Viral replication in the brain causes neuronal dysfunction, leading to the clinical symptoms associated with the rabies disease and eventually death. Early symptom presentation resembles the flu for the first few days, including general weakness, fever, and headache, and can involve discomfort, prickling, or itching at the initial site of infection. There are 2 types of late symptom presentation in rabies: furious rabies and paralytic rabies. Furious rabies can lead to hyperactivity, excitable behavior, hydrophobia, and death within a few days due to cardiorespiratory arrest. Paralytic rabies, which makes up 20% of rabies cases in humans, causes the muscles to become paralyzed, starting at the site of infection and then spreading to the rest of the body. Paralysis leads into coma and eventually death. In other words, the prognosis once symptoms begin isn't very optimistic. However, rabies has been around for a long time, and there are multiple ways to prevent the worst-case outcome. 15 million people every year undergo a treatment called post-exposure prophylaxis, or PEP. PEP is intended to stop the progression of the virus before it spreads to the nervous system, and should be administered within the first 24 hours after initial exposure. PEP consists of thoroughly washing the wound to remove any remaining virus, and injection of human rabies immunoglobulin, or HRIG, and rabies vaccine. HRIG is an antibody that binds to the virus and helps the immune system get rid of it, and prevents its passage into the central nervous system. HRIG is injected at the initial site of infection, while the vaccine is injected far from the infection site in order to speed up adaptive immune response. When administered quickly and correctly, PEP is almost always effective in preventing rabies manifestation. In most cases, pre-exposure vaccination is only necessary for pets or people with a high risk of encountering the virus, such as veterinarians or people researching wild animals or the virus itself. Otherwise, rabies prevention is dependent on proper education and access to medical treatment. With widespread knowledge of and access to PEP, the rabies death toll could be greatly reduced.